All right, welcome everyone to our Build a Cell seminar. It's great to have you here, and I will toss the ball over to Mike to introduce himself and get our talk going. Thank you, Mike.
Great. So just confirm first that you can hear me and see the slide in presenter mode.
Yes.
Or whatever the presentation mode ought to be. All right. Well, thank you for passing me the ball, Carlise, and thank you to the group for the invitation to give a talk today. There was two. I had a little bit of a chat with Carlise as we were getting going. Two very different talks I could give. My lab right now does some work in engineering natural product metabolic pathways. And so the talk that I have queued up right now is more in that line. So if you're engineering multi-gene systems in a cell, what are some approaches for that? I think this is the most relevant to the build a cell community because you could use some of these same approaches to engineer multi-gene pathways in a synthetic cell. The other group of projects that my lab works on is engineering gene flow between sexually reproducing organisms. So how do you create what are in essence synthetic species of a given target organism that can't reproduce with all the wild type versions of that organism? So if there's my guess is that you guys are joining this, you don't know me, you're not going to want to change the direction of the talk. If there's an avalanche in the next few minutes that says we want to hear the synthetic species talk versus this genetic design of experiments, I guess you can write into the comments section while I'm giving the rest of my introduction, and it can be like a choose your own adventure seminar. But with that, I'll say I'm a faculty member at the University of Minnesota. I've been here 8 years now in the Department of Biochemistry and the Biotechnology Institute, which is led by Claudia Schmidt-Dannert, who's on the call as well. For the past 2 or 3 years, I've been highly involved with a manufacturing innovation institute called BioMade. Happy to discuss that with anybody who's interested either on or off the call at a later point. So I'm not seeing many things in the comments, so I'll go ahead and give the genetic design of experiments talk. And I'm going to minimize the place where I could see people's screens if you did have your camera on, which just means if you want to interrupt me, just unmute and shout at me because I can't see you at all. Okay, so one of the biggest needs that I wanted my lab to address when we started here 8 years ago is the need for material, specifically related to natural product drug discovery. This is a figure from a review published close to 20 years ago now, but it's still true today, that there's roughly 3 orders of magnitude more material needs at every stage in drug development. So you can often determine a bioactivity, especially for potent compounds with microgram quantities of material. To determine a structure, it's nice to have a few milligrams, specifically for NMR structure elucidation. Usually you need gram scales to do the medicinal chemistry required for lead development. And to go to a clinical trial, you need anywhere from 100 grams to over a kilogram of material. In having conversations with folks in the drug development industry, I learned that many times, even though genomics-based natural product discovery is a thing now, we can use synthetic biology to turn on metabolic pathways that maybe would be silent in lab conditions and use those to discover new molecules. Usually those molecules are discovered at microgram per liter or milligram per liter scales. And a lot of companies were giving up on really useful scaffolds because they didn't see a reliable route to get to a kilogram of material. And if you can't get to a kilogram of material, their argument was why do all the preclinical development work? And so that's kind of the problem that we set out to address. I have a few slides now that really are maybe non sequitur, but they're going to set the table for what the meat of the talk is about. One is just an introduction to this concept of genetic refactoring. Many of you in this community will have heard of genetic refactoring, where the goal is not to, through the refactoring process, the goal isn't to improve the genetic design of a complex engineered system, but really just to make it more engineerable. So to refactor a natural product gene cluster that would encode a secondary metabolite metabolic pathway, we'd start by removing all the non-essential genes, all of the regulation elements. We would redesign coding DNA sequences, not necessarily to optimize them for expression, but just to enable us to rebuild them using modern tools in DNA synthesis and assembly in thousands to tens of thousands of permutations. We then add back in synthetic regulation to control transcription translation initiation rates and build these back together into an engineered modular multi-gene system. In the talk today, the aspects that I'm going to take advantage of in refactored systems like this are the modularity that allows us to build many permutations in parallel, and the rational control of expression of each gene in the multi-gene system. So the system that we're going to work in, my lab, I push my lab members hard not to just work in model systems, but to work in applied systems. And so this is a compound that we've been working on for a few years now. It's a natural product called seraphendic acid that was discovered in the early 2000s by a group of Japanese neuroscientists. Actually saw Wakana in the audience before I minimize that window. She has probably some funny stories of having to grind up cow livers in her rotation in my lab. But again, for another time. So this compound was discovered by Japanese neuroscientists that were studying rat neurons ex vivo. So they would take rat neurons out of a rat brain, put them onto a plate, and they knew that adding fetal calf serum, or the liquid component of blood from a fetal calf to this plate, would prevent those cells from apoptosing or going through programmed cell death. And so that group in Japan bit the bullet, and from 250 liters of fetal calf serum, which I did the math is equivalent to between 500 and 1,000 fetal calves, they were able to isolate 3 milligrams of this diterpene natural product. They named it seraphendic acid, and they showed that it had nanomolar potency in the bioactivity that they were looking at, so preventing programmed cell death in neurons. That made it really exciting. Programmed cell death is a common part of the pathology of stroke and Alzheimer's, Parkinson's, many neurodegenerative diseases. But interestingly, if you look at the chemical structure, this isn't a mammalian metabolite. Like, mammals don't produce diterpenes with scaffolds like this. And so to this day, nobody knows what the source of that molecule in the fetal calf serum was. Maybe it was in the plants the cows were eating and it was metabolized by liver enzymes. Maybe it's a member of their microbiome is making that compound. But my first graduate student in published work, I'm not going to go into this in detail, thought this was a great target for creating or designing synthetic metabolic pathways. And so since we didn't have a producing organism that we knew made this compound, we had a source. The source is fetal calf serum, but again, we looked in the cow's genome, you couldn't find the enzymes. So instead we just imagined, how would we, if we were a microbe, make a compound like this? And we took genes from different bacteria and combined them to produce this late stage intermediate of seraphendic acid called N-adesaronoic acid. It's only 4 chemical synthesis steps away from seraphendic acid. So we demonstrated a short and efficient total synthesis of seraphendic acid. But importantly, this engineered pathway, which we express in streptomyces, could produce this intermediate at nearly gram per liter scales. So we went from a sourcing issue of having to get 3 milligrams from 1,000 fetal calves to now, working on the gram per liter scale, having access to tens of grams of material that we could do medicinal chemistry on. So the last slide, which is the non sequitur part, this is all the work I'm not going to tell you about. We've used this platform to investigate the mechanism of action, to optimize the synthetic or medicinal chemistry around the scaffold, to improve, find variants that had improved penetration across the blood-brain barrier, et cetera. But the thing that I think is most interesting to this group, and actually it's most interesting to me too, this is kind of the thing that I nerd out on as a scientist, is given in a refactored system, you have the ability to tune relative expression levels of many genes in a pathway. In this case, we had 13 genes in the synthetic metabolic pathway towards N-adesaronoic acid. So given this ability, this capability to independently tune gene expression levels, what's the best or fastest way to optimize all of those expression levels to optimize the titer or yield of a metabolic pathway, for example. So we wanted to draw on 100+ years of work in the design of experiments or multivariate experimental design field that developed mathematical algorithms meant to optimize multi-gene or multivariate systems. Normally not multi-gene systems, because previously we didn't have the tools to go in and manipulate these genetic systems to the degree that one would need to use methods like this. But these same approaches have been really successful in other multivariate systems optimization problems, for example, in chemical engineering or fermentation design, et cetera. So the first approach that we applied to a metabolic pathway is called a Plackett-Berman model. I have an intro to the Plackett-Berman model here. This was first published by Plackett and Berman in the 1940s. And the way that it works is you start by organizing a matrix. Creating a matrix is a little bit like a Sudoku puzzle. Each of the letters across the top of this matrix corresponds to a possible variable in the system. And each number down the side, or the rows in the matrix, are the specific experiments that you would design and run. So the pluses and minuses in this table are because this is the simplest type of Plackett-Berman experimental design, where it's called a two-state experiment design. So with each variable, let's say A is the concentration of glucose, you arbitrarily pick a high state and a low state. So maybe 10 milligrams per, or 10 grams per liter and 15 grams per liter. And the matrix dictates how you combine that variable state with the other variable states to have a balanced Plackett-Berman design. The way that it works is this table is balanced in a way that if you select all of the experiments where one of the variables, let's say variable A, is in the high state. So see in green, I've highlighted 4 of the 8 experiments where A is in its high state. In red, I've highlighted 4 of the 8 experiments where A is in its low state. And if you look at the state of B in those 2 subsets, you'll see that B is balanced. It's got an equal number of experiments where B is in its high and low state. And that allows you to look at the primary effect of variable A, assuming that there's no dependence between the variable states. And that's an important assumption, although I'll come back to that in a second. It allows you to determine the primary effect, or the role of variable A in the overall system performance, by simply summing or averaging the performance of these 4 experiments where A was in its high state and subtracting from that the average or the sum performance of the experiments where A was in its low state. So in a Plackett-Berman design, this property holds true not just for columns A and B, but for any pairwise combination of columns you could draw from this table. And so, you know, it's usually used as a preliminary screen to identify which variable has the largest impact on the system performance. And then you could use another design of experiments to really focus in on that variable and get it set to its optimal state. So we like this. This allows you to reduce what would be a huge combinatorial design space to a tractable number of experiments. It does assume that the variables in the system are completely independent, but it's also been used for things like fermentation media optimization, where that's definitely a dependent system. And an important thing to take as a caveat is in a system that's not completely independent, the dependence between variables, it can exist on a spectrum. So you can have variables that are almost independent, that there's very little interacting terms between variables. And that's the degree of independence of the system is going to impact how well a method like this would work. Okay, I probably beat that to death. One caveat, though, is that we knew that we didn't want to run a 2-state Plackett-Berman. And I can get into that later. Instead, we wanted to run a 5-state Plackett-Berman. So we applied this to the 8 genes in the methyl erythritol phosphate pathway, or the MEP pathway. And for each of those 8 genes, we had the ability to control the gene expression level over about 2 to 3 orders of magnitude with 5 different sets of cistrons. And so we measured these independently and confirmed that we could span that range of expression strength. And the 5-level placket Berman design now is a more complex matrix. Instead of pluses and minuses, I'm showing 5 numerals, 0, 1, 2, 3, and 4. And yeah, I'm only showing the 8 rows of the 8 columns of the matrix that correspond to the 8 variables that we're going to test. But in all, this was a much larger matrix that we drew these 8 columns from. Here it's color coded so you can see there is a pattern to designing a matrix like this. Like I said, it's like designing a really complicated Sudoku problem. But this dictates, in this case, the 125 gene clusters that we would have to build and test to run this placket Berman design in a cell. And this type of genetic design optimization, this type of math would work equally well if you're trying to optimize some property of a synthetic cell. So that's my plug to make it relevant to your group. So Sue, a student in my lab, designed and built these 125 gene clusters. In fact, she did it twice. And then she expressed all of those gene clusters in a model streptomycete host and measured the titer of our target compound and adenosinic acid in replicate in each host. And she saw, actually, before I advance that, I should have pointed out that we saw, just by changing the relative expression strength of these genes, an impact on the overall titer of N-adenosinic acid over about 4 orders of magnitude. So, they all have the same subset of 8 genes in the MEP pathway, an additional 5 genes to convert that to EAA. And only changing that relative expression level had a dramatic impact on the performance of these synthetic gene clusters. So now the exciting part is, like, how do we analyze this data set? Now, one of the straightforward things one could do, this diagram on the right is the assembly diagram for her library. And every line in the diagram shows, for example, that this part, which is a promoter and ribosome binding site combination 4.6, is in, it looks like, 4 or 5 different cistron level parts. Those cistron level parts are combined to partial clusters, and partial clusters are combined to make the full synthetic gene clusters. And each of these intermediate levels of genetic compositions is present in more than one of the full cluster designs. So, for example, we could take a particular motif. In this case, this is partial cluster 37 or something like that, that has, these 4 genes, ISP, D, E, F, and IDI, with this particular pattern of cis-regulatory elements. And we can ask, what's the average titer in all of the full gene clusters that have that motif? This one here turns out to be one of the best motifs. And that's just one way that we could use kind of pattern finding algorithms to, in the next round of design, build, test, keep the patterns that were overrepresented in the well-performing full clusters and throw away the ones that were overrepresented in the poorly performing clusters. To perform an official placket Berman analysis, though, we focus on one variable at a time, and we ask, as we change the expression strength of that variable or that gene, how does that impact overall expression titer? This was a little bit disappointing, to be candid, in that the primary effect sizes weren't that great. We saw a dramatically strong primary effect size for a gene called DXS, or deoxyribulose phosphate synthase, and it was a negative correlation. So for DXS, the stronger we express that gene, on average, less N-adenosinic acid we had in the fermentation. So while most of the correlations on the right look pretty flat, with the exception of DXS, one thing that I didn't mention is when one designs a placket Berman matrix, the typical negative control that one would do in the experiment is to take multiple columns from the matrix, not assign them to any variable in the physical system, and then use those as what we call dummy variables. Because they're all, none of them are linked to the physical system, the primary effect in a completely independent system for each of the dummy variables should be 0. And to the extent that the primary effect for those dummy variables is not 0, is a way to report on how much interaction between variables is there in the system, how much noise in the experimental measurements are there. And so in the bottom left chart here, we're plotting the main effect, which again, we would expect to be 0, and the variation of average titer for, in blue, roughly 20 dummy variables. And when we do it this way, we can see that some of our experimental variables, in fact, I believe 5 out of 8 fall outside of 2 standard deviations away from the subset of negative control points. And so there are slight but statistically meaningful effects that we can detect for multiple genes in the system. But DXS is first gene in the pathway is clearly the most impactful. The fact that the dummy variables do have some variance and they're not all equal to an effect size of 0 suggests that there is interactions of variables in the system. And there's a way that we can quantify that by performing what's called an autocorrelation experiment. So the autocorrelation function is, the equation is shown in the upper right here. To perform an autocorrelation analysis, what one does is looks at the local variance within a small number of steps through a multidimensional landscape like this. So for example, if you imagine from these center points, a distance of 170 in any direction would be covered by these small red circles. And you ask, what's the average variance of the data points that were measured within that distance step? And then how does that variance correspond to the variance of the whole landscape? And so the equation is like this: with a very small distance steps, you expect that the variance in the numerator of this fraction to be very low. And so the overall autocorrelation approximates 1. And as you take larger and larger distance steps, pretty soon the distance that you're stepping will cover that whole landscape. And the variance within the distance step is equivalent to the variance of the landscape. And so this function approaches 0. So you would run this on by taking bigger and bigger distance steps, plotting it like this. And the steepness at which the autocorrelation function drops towards the x-axis reports on what we call the ruggedness of the landscape. So the smoothest possible landscape is what we call a Mount Fuji type landscape. A completely independent system where the optimal setting for any variable in that system is not dependent on where the other variables are set. That would be a completely smooth landscape. It would be the easiest type of landscape to optimize because you could just optimize every variable independently and then combine all those optimal settings and you'd be right at the top of Mount Fuji. That would produce an autocorrelation function that's a straight line from 1 to 0, as you step out in distance. The other extreme is called a Badlands type landscape, where the landscape is so rugged that it drops down to 0 almost immediately. Another way to think of this is that as this autocorrelation drops, it's a measure of, like, let's say you know where you're standing in the landscape, and you want to say, to what degree is the prediction of where I'm standing going to predict, what height I'll be at or what elevation will I be at if I start taking steps in any direction. In Mount Fuji, it's fairly predictive, right? If you're on the side of this mountain and you walk 100 yards in any direction, you're going to be roughly in that same elevation. Whereas in the Badlands, you might be way higher, way lower, you don't know. And so we could run with our data set. We had 125 measured points. We know the expression level or the designed expression level of each of the 8 genes for each of those clusters. And so we can do a simple calculation for Euclidean distance, which looks like this equation up here, except it's got 5 additional terms to the right side of the equal sign to account for the additional dimensions that we're measuring. And so we can calculate a distance, and within our landscape, we have a pretty good spread or range of Euclidean distances between the points. And so when we plot the autocorrelation function, the data is shown in black here. And you can see it kind of corresponds or agrees with what we observed when we did the Plackett-Berman analysis, which is that, yeah, this is a fairly a fairly rugged landscape. So there's evidence that we have interactions of genetic parts in our system, or actually there's another explanation that I'll get to in a couple of slides as well. But it suggests that, you know, this particular pathway is perhaps going to be more difficult to optimize mathematically or predictively without just sampling a lot of places and taking more of an evolutionary approach to improve the performance of the pathway. You know, plotting the data as a network graph where we color code according to the titer of the gene clusters, and we use this clustering algorithm to group things by their distance in this expression space. It kind of tells the same story where, you know, I've false colored the very best performers in green, and you can see none of the top 5 gene clusters is even in the same subcluster. So this confirms again that we have a fairly rugged landscape with maybe multiple local optima, and that can make it difficult to march to the tallest place in the overall landscape. Okay, so the complicated nuance to optimizing this methyl phosphate pathway is that, you know, we came into this thinking about, you know, this is the system. We're optimizing this 8-gene system, and we knew that there is a chance that there'd be interactions between expression levels. We expected there to be interactions between expression levels. We hoped that those interactions would be minimal enough that an algorithm like the Plackett-Berman design would still be effective at identifying the genes with the largest primary effects. Really, though, this isn't the system that we're analyzing. We're analyzing a system that looks more like this, where we're building a synthetic metabolic pathway and having it work in the context of the native metabolism of that cell, which is much more complex. One of the interesting things that we observed, and this is a benefit for the students in the audience of being keen observers of your experiments. Don't just go after the experiments or the hypotheses that you think you're testing, but always keep your eyes open for interesting new directions. When we would introduce these synthetic metabolic pathways to our Streptomyces albifluvitis host, we saw an alarming array of different colony morphologies on the plate. These colony morphologies were you know, classifiable. So there's a classic scheme that one can use in the Streptomyces taxonomy world to classify the phenotype of Streptomyces colonies. And so we use those to group the many different specific examples into 10 different classes of colony morphologies. Each class of colony morphology occurred more than once within our library. And importantly, they would occur reproducibly. So if we, for example, use conjugation to get synthetic gene cluster 47 into the fermentation host, and it produced a colony that looked like this one here in colony class 3, that wasn't just a random event, but if we took that gene cluster and reconjugated it to a naive host, we would get the same colony phenotype 3. And so this is a highly reproducible and unexplained observation. We think that potentially the different pathways that we're introducing are leading to differential accumulation of intermediates or shunt metabolites along this pathway, and that perhaps these isoprenoids are important hormones related to this phenotype of colony morphology that help to control what the colonies look like. And so, you know, we're investigating this a little bit more currently. And so with that, I wanted to acknowledge the students and postdocs working on this project. So this was mostly the work of Susie Sue, who's shown here, and Carlisa. This is sandstone. I did take my lab up there in one of the first few years, but also Dimitri, who's here on the left. I think you guys can see my mouse, and Stephen, who is here in third from the left, Perling. And then Thomas, who's here represented in microbial art. So with that, I will take questions, and I don't have a separate slide for that. So let me make it so I can see your faces again. Are there any questions? If not, this is a short talk already. If there's no questions, it's going to be super short. Someone needs to ask an icebreaker question.
I'm double checking the chat, but I think maybe some people need some time. People like to write in questions to the chat, so maybe it just takes a minute. Let's see.
Sounds good.
Seems like
So
a fun
actually,
group. You've done a
there's
lot
some
of activities.
connections on this project. I'll mention Susie Sue, who did most of this work, was recruited to graduate school by Claudia Schmidt-Danner, who was on the call. Wakana worked on this project a little bit on a tangential project, trying to find some enzymes that that finish the metabolic pathway towards serapenic acid. She had really exciting results once with ground up liver that we had bought, cow liver that we had bought from the supermarket, but we couldn't reproduce it. And so it's interesting in that, like, you know, there's not much information attached to the liver when you buy it from a supermarket. We didn't know, you know, about what genes were expressed in the cow that gave her the positive results in one case, but not the other case. But you know, the last two enzymatic steps are still catalyzed by yet to be determined enzymes.
Interesting.
Hey, Mike, can you hear me?
Yes, Claudia.
So I have a question with the interaction between the proteins that are obviously there. So what about the toxicity or any type of complex formation? How does your design accommodate this? Is there a way to kind of tease this out of the system?
So two ways to answer that. We had toxicity is a problem. The 4 rows that you see in yellow are, no, that's not true. I'm missing, this one here is misplaced. But there were 4 constructs that we couldn't get colonies of. And so one of the constructs, we could never make the gene cluster in E. coli. And it was reproducible. After 3 different attempts to assemble those genes into a construct, something about that genetic design was toxic in E. coli, and we couldn't get past that problem with that construct. And then 3 more, we were never able to get colonies in Streptomyces. In the end, we just said, well, you know, we're testing design on the overall performance. The ability to make the strain is part of performance, so we'll call it 0 titer for those ones. And so yeah, toxicity was an issue. It wasn't in, you know, 4 out of 125 wasn't too much of an attrition rate. I'll say that in terms of how interactions of gene expression levels impacts that toxicity, we didn't investigate that. I can't quite say we couldn't investigate that. There's something called hidden projection properties of Plackett-Berman designs like this that allow for a limited number of interaction terms to be analyzed. Since we didn't know a priori which genes we would want to investigate the interactions of, we didn't incorporate that into the design. You can't look at every possible interaction term. You just, the data set isn't large enough for that. There is the ability, though, to design these Plackett-Berman libraries that you can still determine the primary effects of every variable independently and look at a very small subset of interaction terms. So I can point you to literature if you're interested in doing that, that describe the math behind these hidden projection properties.
Yeah, thanks. I mean, I know that isopenoid pathway is really prone to toxicity and all these additional complications.
Hi, Mike. Can you hear me?
I can hear you, Evan.
Excellent. Beautiful talk. I really enjoy is kind of seeing this really rational approach to biosynthetic gene cluster design. Quick question on how you validate your different designs with other empirical data? Like, are you looking at transcription levels using, RNA-seq? Are you using omics to validate your expectations rather than just looking at titer of your target?
Yeah, we haven't with this library. With a previous work, when we applied similar approach to the nitrogenase gene cluster, we did chase it with every type of omics that were available to us at the time. So strand-specific RNA-seq and proteomics. And the results of the omics in that case largely corresponded with the predictions. I'll say the reason ultimately it would be great to have like omics level investigations of everything. We could still do that with this library, and that would be interesting. There have been in Streptomyces and other microbes pretty robust analyses that look at as you change in combinatorial libraries, as you change, for example, the promoter ribosome binding site interface, or as you change the RBS to coding DNA sequence interface, how much noise does that introduce to, in most of those cases, their reporter genes that they're looking at? So how much noise in reporter gene expression level comes from unexplained variants by changing those genetic parts? And so there was a lot of variance if you change the promoter RBS interface, or like the junction of promoter and ribosome binding site, because the argument would be that the little bit of 5' UTR that's added on by many promoters changes the strength of the RBS. We didn't change that at all. We chose to lock in as the upstream genetic element a combination of promoter and RBS that we measured empirically, each of those combinations. So the only place where we're introducing unmeasured changes in those cis regulatory elements is at the CDS to RBS interface. And the previously published papers have shown that interface accounts for about 13% of the unexplained noise in gene expression levels. So it's not a huge contributor. Like, we expect most of the genes to be expressed at ballpark level that we had measured in the reporter gene system, but we haven't done the omics to confirm that.
Cool. 13% to me seems kind of like a lot, especially when you're dealing with something so finicky, like I guess isoprenoids are somewhat toxic when you do heterologous expression of them. Is there any motivation to reduce that, or is just 13 kind of like the just gonna have to deal with that amount?
There's a lot of motivation. I think that's one really exciting thing. I teach a undergrad lecture here on kind of genetic design, and one exciting thing. So, for example, back to the nitrogenase library where we applied the omics level investigation, one of the things we look for is, can we explain genetic context effects? So why does the positional orientation of different genetic elements impact the behavior or strength of those elements? One thing that we found in that case is that when you have outwardly expressing promoters, like two promoters that are abutting up to each other but expressing in the opposite direction, each promoter is stronger than you would have predicted from the measurements in isolation. And turns out there is papers from like the 70s, one of which showed that if you bind a RNA polymerase to a promoter, it increases the negative superhelicity of the DNA on either side of the promoter. So as you pull the DNA apart, the coil gets tighter on both sides. And a separate paper has shown that if you artificially increase the negative superhelicity of DNA, it actually increases the affinity for RNA polymerase. And so in that case, we could explain that genetic context effects. And actually, there's a paper just published in the last week or two, and I'm drawing a blank on her name, from Galloway at MIT, where she purposely investigated this effect of genetic context. How do you point promoters towards or away from each other, and found the same thing, that superhelicity of the DNA was kind of the driver behind their models. So the reason I bring that up is there's two approaches one could have. On one side, you could try to understand the sources of genetic context effects that right now result in unexplained changes in predicted expression levels. If you understand those fully, you can use those in the designs. For example, if you wanted to boost the expression of a promoter a little bit more, you could put a promoter facing, strong promoter facing opposite from it, right behind it. Alternatively, you could try to build genetic systems that buffer out genetic context effects. And there's been plenty of examples from the synthetic biology field, people using things like self-cleaving ribozymes to insulate between a promoter element and a ribosome binding site element, or different spacer sequences, or bidirectional terminators before and after every cistron to buffer from incoming or outgoing transcription. And so, yes, there's a lot of work. We don't have any active projects in that field. So I think you asked, would there be interest in developing that? I think I'm interested, but not, if I were interested more, we'd probably have projects in that area. But yeah, it's super interesting for me that a genetic design in 20 years might look completely unnatural because it's got all these extra bells and whistles in it just to make it more predictable.
That's really cool. Thank you for sharing. Excellent talk again.
All right, on to lunch. Thank you guys for being here, and have a great day.
Yeah, and thank you again, Mike, for that. Again, wonderful talk. Very exciting, very dense.
Okay, good.
Wonderful. All right.
Take care.